Hey everybody, welcome back to another lecture video. In chapter 20, we are going to be looking at carbohydrates. Now, let's just jump right in. So we're going to go over a little bit of the terminology that we use to describe what we're going to call carbohydrates. Now, I guess we can start with just the term carbohydrate. A carbohydrate technically is a a polyhydroxyaldehyde. Polyhydroxy, right? Meaning that it has a OH, or poly-OHs, many OHs. Aldehyde, remember that's the CHO group, an H on it. Or a polyhydroxy ketone. I guess, or a substance that gives one of these two compounds on hydrolysis, or splitting with water. So if you are a compound that's a polyhydroxyaldehyde or a polyhydroxy ketone, or if you can hydrolyze into one of them, then you're a carbohydrate. Now, monosaccharide is another term that we use. Now, a monosaccharide is a carbohydrate that cannot be hydrolyzed into a simpler carbohydrate. So monosaccharides generally have formulas like CNH2NON, where N can be anything from like 3 to 8. Now, we can also call monosaccharides aldoses or ketoses. Now, an aldose is a monosaccharide that contains the aldehyde group, and ketose is one that contains the ketone group. So here, aldehyde group on the end, remember that's a C double bond O H. And then the carbonyl in the middle here would be the ketose. Now, monosaccharide naming contains suffixes OSE, ose, and this indicates that the molecule is in fact a carbohydrate. In fact, I can probably use this as a better example, right? So an aldotriose, a ketotriose here tells us that it's a carbohydrate. The prefixes, tri, tetra, penta, I was going to say di, but there's no di, sugars, and so forth, hexa, hepta. These will tell you the number of carbon atoms that are inside the chain. And so then, you can combine those together. So if they're aldoses, you can have a triose or a tetrose, or an aldotriose or an aldotetros or an aldopentose. Now there's actually only two trioses being 3-carbon sugars, or 3-carbon monosaccharides. There's the aldotriose and the ketotriose. And usually when you're talking about trioses, since there's only 2 of them, you might find that they exclude the name aldo or keto. So you lose a little bit of information about where the carbonyl carbon is, or the carbonyl group. But it does tell you that there are 3 carbons in it. But again, trioses can be further classified as aldo or keto depending on where that group is. And we could say tetroses and pentoses as well to describe sugars of 4-carbon or 5-carbon number. So that's a little bit of the terminology there. Glyceraldehyde actually is the simplest aldose. And actually these are both glyceraldehyde. And it's the simplest. It's got here 3 carbons, and it's got, this is the aldotriose that we were looking at in the previous example. This is glyceraldehyde. Now it does exist as a pair of enantiomers. It's got a stereocenter, meaning it has a carbon that has 4 different groups on it. And whenever you have a carbon with 4 different groups, there's multiple ways that those groups can be arranged in space. And so you can see here, these are in fact essentially mirror images of each other. Now, we use these representations. not the ball and stick representations on the sides. We use these inner representations called Fisher projections. Now, these are two-dimensional representations for showing the configuration of these tetrahedral stereocenters. I think I have a better picture for you here. The horizontal lines here represent the bonds coming forward from the stereocenter. The vertical lines are the bonds going backward, projecting to the rear from the stereocenter. Only the stereocenter is in the plane of the screen in this case. So these bonds pointing out at you, these going away from you, this guy lined up flat with your screen. Now, this is how the molecule looks in real space. The Fisher projection just has horizontal and vertical lines. We don't need to draw the little arrows. It makes drawing these things simpler. Okay. Now, I'm going to switch these images very quickly. So in 1891, Emil Fisher, the Fisher projection person, arbitrarily assigned the letters D and L to the enantiomers of glyceraldehyde. Now we know that D will basically conform to our R enantiomer, and L is essentially the S enantiomer from our system of our chirality system here. But L and D were just sort of picked randomly. Now, what they mean in the Fisher projection is when we look at the Fisher projection, we look for the, the penultimate carbon, or the last carbon that's chiral. So we're looking for this guy in the molecule, because technically these are the last carbon, but they have two H's on them, so that they're not chiral. So the last chiral carbon, and then we look at where the OH is. And if the OH is on the right, then we call it D, and if it's on the left, we call it L. I'll show you some more examples. Here are some tetroses and pentoses. So tetroses, and these are pentoses. And these are actually D tetroses and pentoses. These are the most common ones. So as far as tetroses go, we have erythros and threose. You can see here the positions of those OH groups on the last chiral carbon. Notice that as these increase from the trioses, the tetroses now have 2 chiral carbons. And the pentoses have 3, and the hexoses will have 4. So every one of these new carbons that we add is just another place to have an L or, not an L or a D, an R or an S configuration. That makes these molecules have many, many stereoisomers. Okay, enantiomers and diastereomers. All right, but the naming is still based on where the position of the last OH is. Here are the most common pentoses, biologically common pentoses, D-ribose and 2-deoxy-D-ribose. These are the sugars that go into your DNA. And then we have our 6-carbon monosaccharides. These are the D-hexoses, and the ones that we're going to talk about. D-glucose, D-galactose are the main ones. There's a 5-carbon sugar that I think I left off of here somewhere, fructose, that we'll look at. It's actually not an aldose, though. It's a ketose, so that's probably why it's not on this list. And D-glucosamine. This is used in some of our biological membranes. Okay. Just some common D-hexoses. You don't have to memorize the names of these. Pay attention to that they're D because of where the OH group is. Now, let's talk about some amino sugars. Amino sugars, in place of an OH, of hydroxyl, have the NH2 group. And so you can see there's a few examples of this here. D-glucosamine, which I showed you in the previous slide, here was a amino sugar. But some of these other ones here, these are still D because of where that group is. All right. Only 3 amino sugars are common in nature. There's the D-glucosamine, there's D-manosamine, and D-galactosamine. Okay. The last one that I've got on here, this N-acetyl D-glucosamine, is actually just an acetylated derivative of D-glucosamine. So this guy comes from this guy. It just has an extra group added, right, to the amino. But there are only 3 common ones. Now, let's talk about cyclic structures. Aldehydes and ketones will react with alcohols to form something called hemiacetals. And I'm going to show you an example of those structures. In fact, you can see a hemiacetal here, but we'll explore it a little better. Aldehydes and ketones, both of them will do this, will react with hydroxyl groups, or we could call it alcohols. Now, all of these functional groups can exist in the same molecule, okay? And that's the case here. So a sugar has a aldehyde group, right? Carbonyl carbon, but it also has hydroxyl groups. And if the hydroxyl group is far enough away from that carbonyl group, we can get a reaction within the molecule. So what you see here, what essentially is going to happen, this O is going to form a bond to this carbon, okay? And this H is going to switch to go up on this oxygen. So the net result is that we formed a ring between the carbons, the O, and that other carbon, and that we've then, that carbonyl carbon is now an alcohol group. And so you can kind of see that here, connect them with a line to show that they formed a ring. This is a 5-membered ring. They typically will form 5 and 6-membered rings. The sugars that we'll look at, the aldoses will look at, form 6-membered rings. The ketose will form a 5-membered ring. And so the hemiacetal is this functional group. It's the carbonyl, well, it's a carbon that has a sort of ether side, right? A C to O, and then O back to another carbon. That's an ether. And that same carbon also has a hydroxyl group. That's a hemiacetal. Hemi kind of means half. Now let's look at what happens when D-glucose does this. So here is the Hayworth projection. Hayworth projection is this sort of ring here. And you can kind of imagine that this ring is laying flat like on a table. And the groups are either above the ring or below the ring, okay? That's the Hayworth projection. So we can take this Fisher projection and we can react D-glucose. So the number 5 hydroxyl will form a bond with that carbonyl carbon of the ring. They will close, the ring will close, and we're going to get 2 what we'll call hemiacetals. One hemiacetal forms with the OH in an up position. The other forms with an OH in the down position. Okay, we call the up one beta, we call the down one alpha. Now, they form randomly, okay? There is a more stable one, and that's the beta form. Beta form sort of exists in more of a percentage if we were to look at a solution of D-glucose. And these things aren't very stable. They open and close, open and close. And so, this molecule can close as a ring and form the alpha D-glucose. Notice that we also have some more fancy names that we can call these, right? This is a pyranose. I don't know if I exactly went over why it's called a pyranose yet, but I'll show you in a second. It has to do with the ring that it forms. There's a furanose and a pyranose ring. But anyways, we can call this thing glucopyranose, but the common name is glucose. And I like to stick to the common names, so don't fixate too much on those fancier names. But again, pyranose here just means it's got 6, that's a 6-membered ring. Now, this can form the alpha D-glucose, and then the alpha D-glucose in water will actually just open up again back into the straight chain. And then the straight chain one will close again, and this time it might form the beta. And then the beta might open up again, and then this time it might form a beta again, and then it might open up and then form an alpha. It's random, okay? But they are constantly going back and forth between the hemiacetal form and the open chain aldehyde form. Now, The 5 or the 6-membered cyclic hemiacetal I mentioned can have different names. And so the new names that we can call these, actually, there was one other term that I forgot. It was the word anomer. Let me go back real quick. So there is the term anomeric carbon, and these two guys are anomers of each other. Okay, they're anomers. It's a type of isomer. It's not really an isomer because their connectivity is exactly the same. It's more of an enantiomer type thing, but the rest of their molecules are the same. It's just the one carbon that's different. And because it was that number 1 chiral carbon, we call that the anomeric carbon. It's what makes these two molecules different from each other. And if you're only different at your anomeric carbons, then you are anomers of each other. So these are anomers. Okay. In the terminology of carbohydrate chemistry, a 6-membered hemiacetal ring is called a pyranose. Because of its resemblance of a 6-membered heterocyclic compound called pyran. So you can probably figure out why the 5-membered ring is called a furanose. It's because it looks like furan. That's all. So furanoses and pyranoses are other terms for 6 and 5-membered hemiacetal rings, and they get their names from these two molecules. Okay, going back to the Haworth projections, aldopentoses also have cyclic hemiacetals. The most prevalent forms of D-ribose, which is one of those pentoses we looked at earlier, are furanoses. They form 5-membered rings. And so here I'm showing you 2 of those. These are the molecules in our DNA, the deoxyribose and the ribose in the RNA and DNA molecules. So again, aldopentoses are 5-membered aldehydes. Hexapentoses are 6-membered aldehydes. The 6-membered ones tend to form the pyranoses, the 5-membered, the furanoses. There is a hexaketose that we didn't look at yet, fructose, that does form a 5-membered ring also. So it's not like it's just one way or the other. In fact, here is that example. So D-fructose is a 2-ketohexose, technically, 2-ketohexose, right? This tells us that it has a ketone group. It's on the number 2. It's got 6 carbons. And this one forms a furanose ring. But of the 3 main sugars that we look at, glucose, galactose, and fructose, these guys are going to make up all of the disaccharides that we tend to be interested in. The maltose, the lactose, and the sucrose, or aka table sugar. So we're going to have to look at how these rings can connect together. While we're on the topic, though, of these pyranose rings, the 6-membered ring, as we've been showing it in the Haworth projection, is actually more accurately represented as a strain-free chair configuration. So what I mean by strain-free is that all the groups are oriented in space in such a way that they're not close to each other or bumping into each other. Conformation. There we go. And so you can see here, we call it the chair conformation because it kind of looks like a chair, you know, kind of like a comfy chair that you would sit in. Anyways, in this configuration, groups can be pointed outwards. Well, there's 2 configurations. There's the axial positions, which are all the up and down positions, and then there's the equatorial positions. And in different arrangements, right, the bulky groups, because every one of these carbons has hydrogens and another group on it. So if you can get all of your bulky groups to be equatorial, they are all sticking away from each other as opposed to being axial. And that can happen if you do a chair flip. This thing can actually flip. I don't think I can draw it. Yeah, I can't draw it. But when they flip, they all switch positions, and rings can do that. Six-membered rings can do this. But anyways, I'm just showing you how we represent more accurately these sugar molecules, okay? All right. And actually in both the Hayworth projections and the chair conformations, the orientation of groups on carbon groups 1 and 5 is always going to be, well, actually, when we're talking about a specific sugar, so this is glucose in both examples, it doesn't change the fact that the groups are going to be the same way. Okay, so whether we look at it in a Hayworth projection or we look at it in the more accurate chair conformation, the groups from 1 to 6, or I guess we could say from 1 to 5, are going to be as follows: up, up, right? They're both, first ones are up, down, down, up, up, down, down, up, up. Now it's easier to see up, down on the Hayworth projection, but on the chair conformation, you have to remember that there's axial and equatorial. So axial was the up versus the down in this case, which would be the axial. And then here we were saying down, and it is down compared to the, actually, I drew that one incorrectly. All the points on these chair configurations are where you should have an up and a down. And then all the other ones are sticking out to the side. This OH is sticking out to the side. But when we compare these two positions, there's an up and a down, and this OH is on the down. And on the number 2 carbon over here, it's also on the down. So all I'm trying to say is here that they're the same. Doesn't matter which conformation we look at, we're going to have the same info. Let's talk about mutorotation. This is a change in the specific rotation that accompanies the equilibrium of the alpha and beta anomers. Okay? When they are, for example, when either alpha D-glucose or beta D-glucose is dissolved in water, the specific rotation of the solution gradually changes from an equilibrium value of about, I don't think it's shown here, plus 52.7 degrees. This is light being passed through the sample, okay? And then it slightly changes, it changes to this. And it corresponds to a 64% beta form and a 36% alpha form. Remember, anytime you put chiral molecules into a solution, they change light a little bit. They rotate plane polarized light. This is what R and S mean. It was the direction in which they rotate that light. What we find when we put, and this is info I already told you that they exist. Glucose, as an example, exists in both the alpha and beta anomers. But this is what this is showing us, is that they don't exist at an equal amount. If they were in equal amount, then they would offset each other. If there was just as much alpha as beta, they would offset each other, and the overall plane rotation of the light would be 0. It wouldn't rotate at all. But because there is a rotation, and we don't have to go through the math or see any of that, so you don't need to worry about the specific equilibrium value. But because we see rotation, we know that this has to do with the amounts, relative amounts of the beta anomer versus the alpha anomer, and there's more beta. What that means for us, though, is that these things turn back into each other. Because what we're starting with is a pure either all alpha or all beta, and instead we get an equilibrium value that corresponds to what we would get if we had a mixture of the two. So mutorotation is just what I told you earlier, that these molecules can open up again back into their straight chain form, and then they seal up again as a hemiacetal, but the OH can be in a different position. So mutorotation is what we call it. let's see. We can treat a monosaccharide with an alcohol to give an acetal. So where we had aldehydes and ketones with alcohols before to give hemiacetals, we can take a monosaccharide, I can't spell, again react that with an alcohol, and then instead of a hemiacetal, we'll get a full acetal. An acetal is a group, a carbon that has two sort of ether groups attached to it, right? COR on one side, COR on the other side. The R here is everything that this O is attached to. The R here is just the CH3. But there was an O attached to both, or two O's attached to that same carbon. Now, the alcohol here, just like the alcohol was the same molecule of the aldehyde and ketone in the hemiacetal formation, this alcohol can be another monosaccharide, another hemiacetal. And that's actually what happens. So if we take beta D-glucose here, doesn't really matter if it's alpha or beta D-glucose, and we react that with, let's say, methyl beta D glucopyranoside. So I guess this is just a methylated version of D-glucose. So it's a methyl glucoside. That's just a term that we don't really need to... Oh, my bad. This reaction is not the two acetals together, or the two hemis. This is still exactly what we were talking about, but instead of, they're giving us small steps first, I'm sorry. So this really is an alcohol, just a little alcohol. So we can make a glucoside, which is a derivative of a monosaccharide, by reacting them with these little alcohols. What they've done here is that they've added, right, a methyl group, essentially, to this carbon, or to this hydroxyl, because it's still a carbon and an oxygen, but now it's a carbon and an oxygen with a methyl on it. And again, that group that formed can still either be up beta or it could be down alpha. It's completely random the way that it happens. Starting with a beta doesn't only get you a beta, you still get a mixture of the two, because once this beta is going to react, it sort of breaks that, you lose that sort of beta-ness about it during the reaction. We're not going to go over the mechanism, but it stops being an anomeric carbon until at the end of the reaction, and then depending on whether it's open up or down is the one you get. So making glycosides is a simpler version of the acetal reaction. Now, I want to show you how we make larger polysaccharides and disaccharides, which involves the same reaction, but we'll be using multiple sugars. Before we get into that, let me see if there was anything else to talk about here. Now, mutarotation is not possible for a glycoside. Unlike a hemiacetal, which is not in equilibrium with the open, a hemiacetal is in equilibrium with the open chain compound, right? That's what we're looking at here, that these are all in equilibrium with each other. It opens and then it closes again, and opens and it closes, and it can be kind of back and forth which anomer you have. But once you get your acetal, it stays in acetal. So mutarotation doesn't happen. So we don't get betas turning into alphas still, okay? These glycosides are pretty stable in water and in basic solutions. Usually in acid, though, these things can be hydrolyzed. Okay. Now, we have compounds called aldetols. Aldetols are essentially sugar alcohols. They're not exactly monosaccharides anymore, or they're not carbohydrates per se, because they lose their aldehyde or their ketone group, right? That's what we defined a carbohydrate as. But they are derivatives of them, and so we get them by reducing, essentially. We reduce the hydroxyl groups by a variety of, we reduce to a hydroxyl group. So the CHO gets turned into basically just a hydroxyl. Instead of this being a C double bond O with an H on it, it becomes that. You lose that double bonded O. And then these just have alcohol groups all the way around them. And these are called alditols, or like I said before, sugar alcohols. These are often used as artificial sweeteners. I think I have a couple more examples of that. D-sorbitol is what we call glucose when it gets reduced. Sorbitol you find on sugar-free gum, packet ingredients, and that kind of thing. Alditols are named by just changing the suffix "-os", like glucose, to itol. And they have common names as well. That's what sorbitol is. But sorbitol is actually also found in berries and cherries, plums, pears, like in the plant world. It's about 60% as sweet as sucrose, which is table sugar. We haven't looked at the structure of that. And it is used in the manufacture of candies as a sugar substitute. Or used in candies because it's sweet, but it's also can be used as a sugar substitute. So it can be used as a low sugar alternative if you're, trying to sweeten something. So again, sorbitol is what we're talking about. There's also these other alditols. I have 3 of them here that are common in the biological world. Only one of these is chiral. The other ones don't have any chiral. The other ones are not chiral because they're symmetrical. So this group and this group are the same on this molecule. Same thing here. These groups are the same. Well, actually, that's not true. I think, no. I don't know which one of these other ones is also not chiral. It doesn't really, maybe because they're symmetrical. Yeah, I don't know. Scratch that. I was going to try to give you a symmetricality example here, but let's not worry about that. But anyways, these are three more alditols: xylitol, mannitol, erythritol. You might again recognize some of these names when you're looking at ingredients. Let's talk about the other reaction of sugars. So they can be reduced to alditols, but they can also be oxidized. We call these aldonic acids. So aldehyde groups, remember ketone groups and ketoses cannot be oxidized, so they don't turn into acids. Only the aldehydes do. So the aldehydes can be oxidized under basic conditions to form a carboxylate anion. The oxidation product, we call it an aldonic acid. And a carbohydrate that reacts with an oxidizing agent to form an aldonic acid, we actually call those reducing sugars. Remember, the reducing agent is the thing that gets oxidized in general chemistry, and same thing with the oxidizing agent itself gets reduced. It just oxidizes something else. So these sugars that get oxidized, they are called reducing sugars. And to be a reducing sugar, essentially you have to be an aldehyde. You can't be aldehyde. You can't be a ketose. You have to be an aldose. You can see that here. So we have D-glucose. Here's our aldose. And the carboxylic acid would have the OH on it. But since we're doing basic conditions when we make these things, these H's get pulled off. And so we end up with, as it said, the carboxylate ion, which has that negative charge on it. This is the carboxylate, remember? Two ketoses can also be reducing sugars, and that has to do with keto-enol tautomerism. So if you remember, we can actually shift around certain double bonds. And long story short, D-fructose, which is a 2-ketose, it can convert into D-glucose in water. And so because that happens, we technically will classify those 2 ketoses as reducing sugars as well, even though they have ketones. But because they They basically rearrange themselves to be aldoses before that happens. The body uses glucuronic acid to detoxify foreign alcohols and phenols. So basically, lets those react with each other. These compounds then get converted into the liver, in the liver to glycosides of glucuronic acid, and then excreted in the urine. Here are two of those examples. Yeah. Oh, well, this guy is the example. This is propofol. This gets converted into this glucuronide. I guess that'd be the word. The glucoside of glucuronic acid, essentially. So here's our glucuronic acid. This was the OH part, right? The alcohol part, foreign alcohol. Actually, this is phenol, the benzene ring with an OH on it. It's still an alcohol. Phenol's an alcohol. This is actually used as an antiseptic. Why are my words not coming out right? It's an anesthetic. That's what I'm trying to say. So the things that knock you out. Propofol. So the way that your body is able to get these things out of your body is it uses glucuronic acid in particular, that particular sugar molecule. The aldonic acid that we were just talking about, glucuronic acid, to convert things into glucosides. And we know the reaction now. That's a hemiacetal reacting with a alcohol to form a acetal. And then this, of course, gets excreted from the body. Let's talk about sucrose. I've been beating around the bush for sucrose. So sucrose is table sugar. We get this from juice of sugar cane and sugar beet. It's got two monosaccharides in it. So it's made up of a D-glucopyranose, or essentially a D-glucose. It needs to be alpha D-glucose specifically. And then it's also made up of a beta D-fructose, or fructofuranose. And the fructose we didn't really look at, but this is fructose down here. And fructose is only different from glucose in that it has a ketone group instead of an aldehyde group. So on the 1 and 2 carbon, the double bond is just slid over. So it forms a slightly different ring, right? When it makes its hemiacetal, it forms a furanose ring as opposed to a pyranose ring. That's why they look different. And then again, it takes the specific alpha version, which has its OH group down, and the beta version of this guy with its OH group up to do a reaction. With these reactions, we call them glycosidic bonds. And the naming here, you can kind of see that it's explaining which carbons are reacting to which carbons, or bonded to which carbons. Here it's telling us that we have an alpha 1,2 glycosidic bond. So it's alpha because it's down from this guy. Alpha 1, 2. It's the 1 carbon of this guy to the number 2 carbon here. The number 1 carbon is actually this guy right here. And glycosidic bond. We have a few more to look at. This is the strange looking one because it has that 5-membered ring. But the other ones have 6-membered rings in them. We have lactose. This is the principal sugar in milk. It's about 5 to 8% in human milk, 4 to 5% in cow's milk. Here they are connected with a beta 1,4 glycosidic bond. So where the other two sugars were connected on their anomeric carbons, this one only has one anomeric carbon. This particular glucose, actually this is galactose. This is beta galactose. And beta galactose is reacting with a beta glucose. And it doesn't have to be a beta glucose, it could be an alpha glucose, because it doesn't really matter. The glucose, right, is the second membered in the ring. The position of this OH group could be up or down, and that doesn't change anything for lactose. In fact, while this thing is hooked up as this disaccharide, which is technically what we call two carbohydrates that are linked together, instead of mono, meaning one, di for two. This side of the ring, the blue one, still does open and close, and we get the beta or alpha-ness flipping back and forth. So that's that doesn't matter here. But in terms of which ones that react together, we definitely need a unit of the beta galactose because that has the OH in the correct position to react with the down hydroxyl group. That's in our, of course, this is not the one. I shouldn't have circled that guy. Sorry. To react with the down OH, just to kind of give you a better picture. So, right, 1, 2, 3, 4 has a down OH. That's the glucose, and then this one has a up OH. So that's why this one has to be beta, so that they can form a bond. They're the right orientation. Doesn't matter what's going on over here. So we call it a beta 1,4 because it's beta from the 1 carbon of galactose to the 4 carbon of glucose. So it's a beta 1,4 glycosidic bond. One more of importance, maltose. This is what we get when we take glucoses and mix them together. We can do alpha glucoses here. Alpha glucose, alpha D glucose. They're all Ds when we talk about biological sugars. So alpha glucose here forms a alpha 1,4 glycosidic bond. So it's the exact same thing that's happening with lactose, except in lactose this wasn't a glucose; it was a beta galactose. That's the only difference. And they still connect at the same 2 carbons, the 1 and the 4. The bond, of course, in this one is called an alpha because the OH group was down on this sugar and down on the other sugar. So it was like this. So these guys are linking together. We lose a water. I forgot to mention that. There is an H and an OH, right, from each side. But at the end, there's only one oxygen in between them. So we lose a water molecule in these linkages. And that's not actually being shown here as products. So maltose comes from just linking glucoses together. Now our body and plants and anything that's storing starch or sugar or that kind of stuff uses the alpha 1,4 glycosidic bond. If we were to start using beta 1,4 bonds using glucose molecules, we actually get a much different molecule. I'm going to show you that in a couple of minutes. So let's talk about polysaccharides. So the first polysaccharide of importance, well, first let's just talk about what it means. Carbohydrates that have a large number of monosaccharide units joined together by these glycosidic bonds are called polysaccharides. Starch is a polymer of D-glucose. And this right here is an example of starch. Starch can be separated into 2 kind of categories. We have amylose and amylopectin. Now, amylose is composed of unbranched chains up to about 4,000 D-glucoses long, all linked by alpha 1,4 bonds. Now, amylopectin contains chains up to 10,000 glucoses. It's also joined by alpha 1,4 bonds, but they have branches every 24 to 30 units or so. There's a branch at an alpha 1,6, and so you can just see a small segment of that here. So amylose and amylopectin are the 2 different sort of polymers that you can find in starch. Now, different starches have different ratios of these. Some starches have more amylose, some have more amylopectin. But anyways, these are the 2 differences between those. Now, glycogen, I guess I'll stay here. Glycogen is what we have. So what animals use. Now, this is, again, plants' energy. Now let's talk about animal energy. Glycogen is a branched polysaccharide, again, of approximately 10 to the 6 glucose units. So of about 1,000,000 glucose units. Again, alpha 1,4s and alpha 1,6s for the branches. Okay, the total amount of glycogen in the body of a well-nourished adult human is about 350 grams, divided almost equally between liver and muscle. There's way more branches, way more glucose units, and this just means faster energy, more energy quickly. Because when we are breaking down glucose to use, we clip it off of the ends. It gets clipped off of the ends of these long chains and used. So the more branches you have, the more ends of chains you have. And so why do you think animals are going to need more energy quickly than plants? Because they have to run away and hide and do all kinds of stuff, and plants don't. All right, cellulose. This is a linear polysaccharide of D-glucose, also joined by beta 1,4 linkages. Again, D-glucoses here. Average molecular weight of about 400,000 grams per mole, which is about 2,200 glucose units. So smaller than than all of them, basically, right? What did we say it was? About 4,000 even for just starch. So a lot less of a molecule, cellulose. But these molecules act like stiff rods. And they align themselves side by side into these well-organized water-insoluble fibers. And then the hydroxyl groups form all these kind of interconnected hydrogen bonds. And this arrangement of these like parallel chains and these bundles, right, all going back and forth like this, all hydrogen bonding and linking, and makes a really strong mechanically strong, okay, fiber. It's also the reason why it's insoluble in water. So you haven't figured out what we're talking about yet. We're talking about cotton. Cotton is cellulose. It's sugar molecules strung together for structure. Plants do this, right? Instead of for storage and for energy. These things don't dissolve in water. They make really good textiles. So here is that structure of cellulose. Now, let's see. Humans and other animals, as you probably figured, can't digest cellulose. And this is actually because we don't have the right enzymes. We have enzymes called glycosidases that hydrolyze alpha 1,4 bonds and alpha 1,6 bonds, but not beta 1,4 bonds. You would think that that'd be an easy thing to do, but enzymes can't do it. Termites and other organisms that like termites, funguses, that do eat wood and cellulose, have bacteria usually in their guts or in their intestines that can actually digest the wood. Ruminants like horses and cows can digest grasses and hay because they have, again, these organisms in their intestines that can, these bacteria that have the enzymes to break those down. Now we only have the alpha glucosidases, so we can't do those. But lots of bacteria have those, and microorganisms in general have those beta glucosidases. Lastly, let's talk about some acidic polysaccharides. There's a group of polysaccharides that contains carboxyl groups and or sulfuric ester groups, and here you can kind of see those sulfuric ester and carboxylic acid groups or carboxyl groups. And these play an important role in the structure and function of connective tissue. There's no like single general type of connective tissue. There's a large number of highly specialized forms like cartilage or bone, synovial fluid, skin, tendons, blood vessels, and so on, right? But most connective tissue is made up of collagen. It's kind of a structural protein, and in combination with a variety of acidic polysaccharides. So that's why we're talking about it. So heparin, which I've actually got shown here, is synthesized and stored in mast cells of various tissues, including like the liver, lungs, and in your gut. It's best known and understood of its kind of anticoagulant activity. It strongly binds to something called antithrombin 3. It's a plasma protein that's involved in terminating the clotting process. And, it's just an example of a pentasaccharide. This is a repeating unit of the pentasaccharide. This is a pentasaccharide repeating unit. So more of these 5 groups, at the beginning and end. of heparin. So you can just kind of see what heparin looks like. And heparin, again, is an acidic polysaccharide. We could say that this is a beta 1, 4, beta 1,4 bond, right? Now that we know how to name these things, this one up here is an alpha 1,4 bond. And again, these are not regular sugars, so we're not trying to break these down for energy, but just noting these glycosidic linkages, we can sort of puzzle these things out. Anyways, that's the end of this chapter. Again, acidic polysaccharides are not the focus of this, but just an example of how our bodies use similar molecules to do different jobs. All right, guys. I will see you in the next video.